On the 21st November 2014, a camera bolted to a baited lander on the floor of the Mariana Trench filmed a fish. The lander was part of the Hades program, Hadal Ecosystem Studies, operated from the Schmidt Ocean Institute's research vessel, Falkor. The footage is publicly archived, depth labelled at 7,949 meters, and attributed to a real scientific expedition. The Schmidt Ocean Institute has made this footage accessible, and it appears in multiple peer-reviewed publications with full attribution. What it shows is a small, pale creature drifting past the bait with a kind of unhurried ease, barely more than 20 centimetres, pink-white, apparently boneless. That fish was later formally described as Pseudoliparis swirei, the Mariana snailfish. Between 2014 and 2017, 37 individuals were collected via the Hades program at depths ranging from 6,898 to 7,966 meters. In November 2017, Mackenzie Gerringer, Thomas Linley and colleagues published the species description in Zootaxa, recording the following about what the fish looks like in life. The skin and peritoneum are transparent, and the internal organs, the liver, the stomach, the muscle bands along the trunk, are clearly visible through the skin and the thin abdominal wall. A 2019 genomic study by Kun Wang and colleagues, published in Nature Ecology and Evolution, put it more directly. Its skin is so transparent that its muscles and internal organs are clearly visible through the skin and abdominal wall. This is what creates the problem. A fish with thin, nearly transparent skin, an incompletely ossified skeleton, and a body composed largely of a gelatinous tissue that is more than 95% water, holding the confirmed record for the deepest fish ever caught on the seafloor. 7,966 meters. If a fish were ever designed to look like it couldn't survive, this is the one. If a human could breathe underwater, could they survive at the depths P. swirei inhabits? The question sounds like a thought experiment, but the answer comes quickly. No. Breathing is only one of several environmental challenges, and not the most immediately lethal at extreme depth. The human body is mostly water and relatively incompressible. Solid tissue does not behave like an empty container under pressure. What fails first are the air-filled spaces, the lungs, the middle ear, the sinuses. These follow different physics from solid tissue. The chest wall and lungs reach a mechanical limit at around 30 to 40 meters, beyond which they can't safely compress further without rupture. At the pressures of the Hadal zone, approximately 800 atmospheres at P. swirei's confirmed capture depth, no unprotected human would survive long enough to encounter the seafloor. The failure isn't an implosion of solid flesh. It's the collapse of air spaces, and it's fast. P. swirei has no air spaces, no swim bladder, no rigid enclosed cavity designed to maintain internal pressure at odds with the environment outside. Its relevant adaptations are structural and biochemical. 3 of them, each counterintuitive in a different way, go some distance toward explaining how an animal that looks so fragile is in fact precisely suited to the one environment on the planet least likely to tolerate biological delicacy. The first is the skin. The 2017 species description records the following observation directly. Skin thin, transparent, subdermal extracellular matrix thick below skin and between muscle bands. That is careful scientific language worth unpacking. The outer skin layer is described as thin, but there are no published numerical measurements of skin thickness for P. swirei, no micrometer scale figures in the peer-reviewed literature. What exists is qualitative description from multiple independent teams and quantitative confirmation of the transparency itself. A 2026 study by researchers at Tsinghua University, applying an automated computer vision pipeline to lander footage from the Mariana Trench, derived a transparency index of 94.2% for P. swirei swimming freely at depth. The transparency is a confirmed live animal trait, not damage from decompression or surface handling. The 2019 Wang et al. Genomic study adds a genomic anchor. P. swirei has completely lost the pigmentation gene designated MC1R, along with several photoreceptor genes. The transparency isn't incidental, it's encoded in the genome. This fish can't produce pigment. Across generations of Hadal life, the selective pressure to maintain color in a place of permanent darkness has disappeared, and the gene that would have produced it has been lost entirely. Why thin skin? This is where the science becomes deliberately cautious. The gelatinous subdermal layer beneath the thin outer skin is less dense than seawater, which may contribute to buoyancy, a significant advantage for a fish at depth with no swim bladder. The thin skin may reduce the energetic cost of building and maintaining a large body. Some technical literature describes a mechanism by which water-permeable tissue allows internal and external pressure to equilibrate, but this specific causal link hasn't been experimentally confirmed. What scientists can say with confidence is what is observed: thin, transparent outer skin over a thick, gelatinous subdermal matrix, the composition and floating properties of which are directly measured. What remains hypothetical is the precise mechanical relationship between skin thickness and pressure tolerance. The observation is settled, the mechanism isn't. One more piece of context changes the framing. Thin, soft, gelatinous skin is a baseline trait of the Liparidae family. Snailfishes generally, from tidal pools to polar seas, share this body plan. P. swirei didn't invent softness under pressure from nothing. What the genomic data indicate is that the Mariana snailfish has taken a pre-existing family tendency and amplified it. Complete pigment loss, the most extreme recorded transparency in the family, and structural features not observed in shallower relatives. The Hadal environment didn't create the softness. The evidence suggests it selected for an extreme version of it that goes well beyond anything found in coastal snailfishes. The second adaptation is the skeleton, or more precisely, what the skeleton lacks. Wang et al. Described what microcomputed tomography of P. swirei reveals. The skull isn't completely closed. Most of the bones consist of cartilage rather than being ossified. The skeleton is incompletely ossified, and the bones are thin. The genomic analysis identified a frameshift mutation in a gene that regulates bone mineralization and cartilage calcification. When researchers disrupted the equivalent gene in zebrafish embryos, staining for mineralized tissue showed measurably reduced skeletal development. The frameshift mutation in P. swirei may be associated with the species' incomplete skull structure and reduced bone hardness. The word the 2019 paper uses is may, and that precision matters. A 2023 chromosome-level genome study by Xu et al., published in eLife, compared the skeletons of Hadal and shallow-water snailfishes directly using micro-CT. The results showed shorter bones and reduced bone density in the Hadal snailfish compared to Tanaka's snailfish, a shallow-water relative. The authors note this conclusion is based on limited samples and requires further verification. A 2021 study by Gerringer and colleagues, examining skeletal morphology across the full snailfish family, confirmed the pattern of declining bone density with increasing habitat depth, but stated explicitly that skeletal changes are driven not only by hydrostatic pressure, but by other environmental factors and evolutionary ancestry. Pressure is likely one selective force among several, so the popular claim that the fish has no bones is inaccurate. It has bones, thin, incompletely ossified, substantially cartilaginous bones. The distinction is worth preserving because the accurate observation is actually the more interesting one. A rigid, fully enclosed skull in an environment with pressure differentials of hundreds of atmospheres presents structural challenges. A skull with open spaces, permitting internal and external pressure to equilibrate, is a different architecture entirely. Flexibility under compression rather than rigidity against it. The gelatinous tissue beneath the skin is more than 95% water, directly measured. Samples of this tissue from related snailfish species float when placed in cold seawater, lower density than seawater, providing positive buoyancy without a swim bladder. Gerringer's 2017 research into the gelatinous layer included a robotic snailfish model with a water-filled tail, which swam more efficiently than the same model without. The function of the gel may be multiple and simultaneous: buoyancy, body profile maintenance, swimming efficiency, and metabolic economy. A cheap way to build body volume at depth where food is scarce and energy budgeting is relentless. What these two sections share is a structure. Here is what is observed. Here is what scientists hypothesize it does. Here is how confident the evidence makes us. The open skull and the mutation in the bone mineralization pathway are the most directly documented. The buoyancy contribution of the gelatinous layer is supported by floating experiments and comparative anatomy. The rest sits at varying confidence levels. Gerringer and colleagues are consistent about not collapsing those levels into a single, tidy explanation. The third adaptation is invisible. It wouldn't appear in any lander footage, any micro-CT scan, any specimen photograph. It operates at the level of molecules, and it's arguably the most thoroughly documented of the three. Proteins fold into specific three-dimensional shapes, and those shapes determine their function. High hydrostatic pressure forces water molecules into protein structures, distorting the folding and degrading function. The proteins that work in a shallow water fish at atmospheric pressure would, at 800 atmospheres, lose structural integrity and cease to function normally. Deep-sea organisms have two broad strategies to address this: intrinsic changes to protein sequences that make those proteins more resistant to compression, and the accumulation of small organic molecules that stabilize proteins against pressure-induced distortion. The second strategy has been documented with precision across decades of research. Paul Yancey, whose work at Whitman College has built the foundation of this field, published experimental evidence in 2001 in the Journal of Experimental Zoology, showing that a specific class of these stabilizing molecules can fully offset pressure-induced changes in enzyme kinetics, and can completely counteract pressure inhibition of protein assembly. These aren't theoretical projections. They are measurements from controlled laboratory experiments. In 2014, Yancey and colleagues published a synthesis in the Proceedings of the National Academy of Sciences showing that concentrations of these stabilizing molecules in teleost fish muscles increase with depth across 17 species from 9 families, from relatively low levels in shallow water to nearly 400 millimoles per kilogram near 8,000 meters depth. The correlation has an R-squared value of approximately 0.91. The 2019 genomic study of P. swirei identified expansions and mutations in the gene pathways responsible for producing these molecules, consistent with increased production in a Hadal environment. Research from the Woods Hole Oceanographic Institution, citing Gerringer directly, states that P. swirei produces these stabilizing molecules to help maintain protein function under high pressure, describing them as essential for the fish to survive at these depths. Direct biochemical measurement of the concentration in P. swirei tissue specifically hasn't yet been published, but a related Hadal snailfish, Pseudoliparis kermidescensis from the Kermadec Trench at 7,000 meters, has been measured at 386 millimoles per kilogram, approaching the upper range documented for any fish. That figure connects to something that makes the story more precise and more honest. The same 2014 PNAS study proposed that concentrations of these stabilizing molecules above a certain threshold would make fish cells osmotically equivalent to seawater, creating fluid balance problems that would likely become lethal. The modeled threshold corresponds to a depth of approximately 8,200 meters. The confirmed capture depth of P. swirei is 7,966 meters. A snailfish filmed in the Izu-Ogasawara Trench in 2022, a Pseudoliparis of undetermined species, not P. swirei, not the Mariana Trench, holds the current depth record for any fish at 8,336 meters, but it was filmed, not caught, and the species hasn't been formally confirmed. P. swirei isn't simply thriving in the Hadal zone. It's operating close to the theoretical biochemical ceiling for fish life. The adaptation that permits it to function at these depths is approaching the concentration at which it creates its own physiological constraint. The fish isn't infinitely adapted. It's finely adapted, right up to the edge. If you have played Subnautica, some of this will look familiar at a distance. The game's translucent, alien-looking deep-water creatures share an aesthetic logic with the real biology. Bodies built for darkness and pressure, moving with indifferent ease through environments no surface creature was designed for. The community comparisons aren't accidental. Real Hadal fish are small, harmless and fragile-looking, the inverse of Subnautica's predators in every way except appearance. But the visual grammar is the same. Translucency is what depth looks like when you strip out pigment, scales, and the architectural assumptions of sunlit water. The deeper principle here is what makes all three adaptations cohere. The more radically different the environment gets from our own, the more radically different the adaptations to live there need to be. That isn't a philosophical statement. It's the measurable outcome of natural selection acting on an organism across roughly 20 million years. A human engineer designing a vessel for Challenger Deep begins by asking how to make something strong enough to resist compression. The logic is rigidity, reinforcement, enclosure. The bathyscaphe Trieste, which first reached the bottom of Challenger Deep in 1960, had walls built to keep the inside and outside isolated from one another. That is the only engineering solution available to a surface-built machine operating in a surface-built logic. P. swirei works from the other direction. No swim bladder to implode. No rigid enclosed skull to fracture under differential pressure. No pigment to maintain in a place without light. A thin outer skin over a thick gelatinous matrix. An open skull. Molecules accumulating in its cells at concentrations that would be unusual in any shallow water fish, holding its proteins in working order at conditions that would denature those same proteins in a surface species. And a body composed of tissue so watery, so lacking in structural density, that a 2026 automated video analysis found it to be 94.2% transparent in free-swimming footage at depth. What looks like structural inadequacy is structural precision. Each apparent fragility, the transparent skin, the incompletely ossified skeleton, the invisible molecular chemistry, is calibrated to the specific demands of an environment 7,000 meters below the surface. Not every causal link is fully confirmed, not every function is definitively proven, but the convergence of these observations across genomics, morphology, biochemistry, and in situ video points consistently toward the same conclusion. The fish isn't less built than a surface fish. It's differently built, for a set of physical conditions that has no equivalent in the world where human intuitions about strength and weakness were formed. Apparent fragility isn't biological inadequacy. Bodies shaped by radically different environments don't need to resemble what humans have learned to call strength. Which of those three adaptations, the transparent skin, the incompletely ossified skeleton, or the invisible molecular chemistry that holds its proteins in shape, is the hardest to believe is real? Leave your answer in the comments.